Dear students, welcome to SETI Botany classes. In this video, we will discuss what are the various tools of recombinant DNA technology. Now, in recombinant DNA technology, we need to have DNA of particular sequence, particular characteristic feature. That DNA is cut with the help of enzymes. In this video, we will emphasize on this part what are the various enzymes which are involved in cutting of the DNA. Then that cut DNA is inserted into the host or the target tissue. And then that DNA is recombined with the host or the target tissue. And that will produce a new characteristics. This is called as recombinant DNA technology or genetic engineering. For this whole process, we need to have various important tools. And what are the various tools which are involved in the whole process? Number one are various kinds of enzymes. Enzymes play an important role in cutting the DNA from a particular point, and then that cut DNA is multiplied and cloned with the help of carriers or vectors. So we can make multiple copies of desired DNA with the help of carrier or vectors. Then that carrier and vector is recombined with the foreign DNA. Then that is again used with the help of probes. Probes are DNA and RNA. DNA is labeled, RNA is labeled, and this is called as probes. And there are linkers. This we'll discuss in the later videos. So there are tools. Number one, enzymes. Number two, carriers or vectors. Number three, foreign DNA. Number four, probes. And number five, linkers. In this video, we will emphasize on various enzymes which are involved in genetic engineering or recombinant DNA technology. Now, if we talk about enzymes, first we'll discuss lysing enzyme or lysozyme. These enzymes can degrade bacterial cell wall, open up the bacterial cell wall, and then these enzymes, when dissolve the bacterial cell wall, they're causing lysing. So they are called as lysing enzymes or lysozymes. Then second category of enzymes are cleaving enzymes, or we call them as nucleases. Now, nucleases are enzymes which will degrade or act on nucleic acids. Now, nucleic acids are of two types, RNA and DNA. If these nucleases, they act on RNA, they are called as RNAs. So they will break open the single-stranded RNA. As we know, in DNA and RNA, its structure is made up of nitrogenous base, phosphoric acid, and sugar. And then RNAs will break the bond between sugar and phosphoric acid. And that bond is broken, then RNA is also broken. So this is called as RNAs. Similarly, when these enzymes act on DNA, they are called as DNAs. These enzymes will also break DNA molecules by hydrolysis of its sugar phosphate backbone mean. The bond between its deoxyribose sugar and phosphoric acid is broken. In case of RNA, bond between ribose sugar and phosphoric acid is broken. So number one, enzymes are lysing enzyme. Number two are cleaving enzymes. Cleaving enzymes include nucleases. If they cut on RNA or break the RNA or degrade the RNA, they are RNAs. And if they degrade DNA, they are DNAs. Now, nucleases are further classified into various categories. If they cut DNA in the terminal region, they are called exonucleases. And if they cut the DNA in the middle region, they are called as endonucleases. So endonucleases are further, if they act on specific sequences, then they are called as restriction endonucleases. This we will discuss in the second video. So specific endonucleases, which cleave the DNA at specific sequences, are called as restriction endonucleases. And when they cut anywhere on the DNA in the middle region and that breakdown of DNA is random, this is called as non-specific endonucleases. cleaves DNA at random sites. So we have exonucleases and we have endonucleases. Endonucleases are further if they are non-specific, cutting DNA at random site, and they may be specific, cutting at specific sequences. Now we'll talk about exonucleases. Now this I've shown the DNA. One is 5' end, another is 3' end. Here it is 5', then again 3' end. So these enzymes, when they cut on the outer side toward the 5' or toward the 3' end, they are called exonucleases. So these enzymes attack the nucleic acid at their terminal sites only. They act either at the 3' end or 5' end. So they are further of three types: exonuclease 1, exonuclease 2, and exonuclease 3. Exonucleases 1, these enzymes will act only on the single-stranded DNA, and exonuclease 2 will act on the double-stranded DNA, and they will not attack the RNA. Then we have exonuclease 3. They are specific for the double-stranded DNA. They will attack the 3' terminal end of the DNA. That end may be free or phosphorylated. Phosphorylated means when phosphoric acid is there, this is called as phosphorylated. If phosphoric acid is removed, then we call that end free. So exonuclease will act on the terminal ends of the DNA. They're categorized into three categories: exonuclease 1, exonuclease 2, and exonuclease 3. Then we have endonucleases, which will cut the DNA in the middle region. Like we discussed, if they cut randomly, then they are non-specific. And if they cut at specific sequences, then they are called as restriction endonucleases. So these sequences recognized by the DNA are highly specific. So these are also called as palindromic sequences. This we'll discuss in the next video. If you like my video, please like, share, and subscribe. Thank you for watching my video.